So welcome everybody to this lecture on DNA structure and replication. So this is chapters 10.1 to 10.5 in your book. So here are really the major points for the next two lectures. So today we're going to be focusing on the structure of DNA and RNA and also on DNA replication. Next time we're going to talk about how exactly we get from DNA to proteins. So through the processes of transcription and translation. So next time we'll focus on those two processes, but today we're going to be talking about the structure of DNA and RNA as well as DNA replication. So to help you guys remember what exactly a DNA nucleotide is, here's a question that's very similar to the ones you had in chapter 3 when we focused on biomolecules. So which of the following is a DNA nucleotide? A, B, C, or D? So here the correct answer is B. So remember, a nucleotide has three major parts. It has a phosphate, a sugar, and the nitrogenous base. So if you look for those three parts, you'll be able to easily identify a DNA nucleotide. So A, just for your information, is actually an amino acid, C is a sugar, and D is actually a steroid. So that leaves us with B. So B has the three major parts of a DNA nucleotide: a phosphate group, a sugar, and an nitrogenous base. And here's that just again in a blown up figure. So you can see we have a phosphate group, a sugar. Depending on DNA versus RNA, that sugar may either be deoxyribose or ribose. And then we have some sort of nitrogenous base. So nitrogenous bases, there are actually 5 of them. There's adenine, cytosine, guanine, thymine, and uracil. So we'll talk a little bit about the structures of nitrogenous bases later in this lecture. So as you guys all know, DNA forms a double helix. So this is the very common structure you've probably seen in other classes. So DNA is the heredity molecule for cells. So it's a very important molecule. So as we've talked about in other lectures, DNA codes for proteins, and those proteins then do the variety of work that your cell needs to accomplish. So that makes DNA a very important molecule for cells and life. So here we're zooming in on one of those strands of a DNA molecule. So as you can see, they have various parts. So we have this part right here, which is called the sugar phosphate backbone. So nucleotides of DNA are actually linked together through the sugar phosphate backbone. And perhaps the easier figure to look at is the one on the right. So you can see we have phosphate groups linked together to sugar molecules. And that pattern just keeps on repeating and repeating. So we have phosphate and sugar molecules linked together. So it's important to know that, that the sugar phosphate backbone is linked together through the sugars and phosphates. It's not the nitrogenous bases that are linking together. So if you look closely, it's sort of alluded to, but we have covalent bonds linking these molecules together. And more importantly, actually polar covalent bonds are linking them. So we'll talk a little bit more about why this is important when we do our DNA extraction lab. So the fact that we have polar covalent bonds on the outside of DNA strands is very important for the DNA extraction procedure we're going to be doing in lab. Okay, so we talked about the sugar phosphate backbone, so that links the nucleotides of DNA together. Next, we're going to talk about nitrogenous bases. So as you can see, we have nitrogenous bases hanging on the outside. So whereas the sugar phosphate backbone linked nucleotides together on the same strand, the nitrogenous bases actually linked together the different strands of DNA. So if I go back to the previous slide, you can see that the nitrogenous bases are interacting. So we have A's matching up with T's, C's matching up with G's. So C, G, and AT, I should say, are the different nitrogenous bases. So C is cytosine, G is guanine, A is adenine, and T is thymine. So we'll talk more about those later in the lecture. But you can see that these nitrogenous bases are interacting, and that's what's linking up the two strands of DNA. More specifically, we have hydrogen bonds interacting between these two. So remember, the sugar-phosphate backbone is polar covalent bonds. So that's linking together nucleotides of the same strand. When you want to link together nucleotides of different strands, though, that's hydrogen bonds between the nitrogenous bases. So please be aware of that. It's very important that you remember the different types of bonds in DNA. So here's that all together. So we have a double helix here of DNA. Looking at one of those strands, we have a sugar-phosphate backbone that's linked together through polar covalent bonds. Each of those nucleotides has its own nitrogenous base. So A, C, T, G, G here. And if we had another strand of DNA, you could see that those nitrogenous bases are actually hydrogen bonding with the opposite strand. Looking at a single nucleotide, again, the major component is a phosphate group, a sugar. In the case of DNA, that's deoxyribose, that's the name of the sugar. And then we have our nitrogenous base. So essentially, what I've been talking about is essentially zooming in on DNA. So when we look more closely at the sugar, we actually have a naming scheme for the carbons that are found in it. So the carbon that's closest to the nitrogenous base, we call the 1'. The second carbon is called the 2', third carbon 3', 4 carbon 4', 5 carbon 5'. So essentially it goes clockwise from the nitrogenous base. So this is going to be very important in later slides when we talk about directionality of DNA. So the naming of carbons, in this case the 1', 2', 3' system, allows us to tell the direction of DNA, which is going to be very important. As I just alluded to, DNA has a direction. And those directions specifically point to the carbon that is unlinked. So if you look here, we have the 5' carbon is unlinked. So we have a lone phosphate group there that's attached to nothing. So that is the 5' end of DNA. Here on the other strand, though, we have a 3'. So that 3' is alone by itself, so it just has a lone hydroxyl group, so that's not connected to anything. So that's the 3' end of DNA. If you look at the other end of the left-handed strand, you can see we have a lone hydroxyl group again. So that lone hydroxyl group is on the 3' carbon, so that gets the name 3' end. If you look on the other strand, we have a lone carbon there that's attached to a phosphate group that's not attached to anything else, so we call that the 5' end. So as you may have figured out already, DNA strands run anti-parallel. So if their 5' end is over here, then it must be the 3' on the other end. So you can't have 5' on both ends of DNA. So if you travel down the left-hand DNA, the other end must be 3'. And if you know that the other end is 3', since DNA is anti-parallel, the other side must be 5'. So we'll have some opportunities to practice this in later slides. So here's just a review. So what prime carbon is indicated by the arrow below? So here we have the 5' carbon. So E, so remember it goes clockwise from the nitrogenous base. So this is 1, 2, 3, 4, 5. So that's the 5' carbon indicated by the arrow. So here's a little bit more tricky of a question. So is the arrow pointing to the 5' or 3' end of the DNA? So here is actually indicating the 3' end. So here we would have a lone hydroxyl group. It's not written out for you, but it's implied. So these circles right here are actually the phosphates. And these sugars right here are the deoxyribose. So it's important to point out here that deoxyribose is also called a pentose. So pentose sugars form pentagons in their circle form. So let's just be aware of that. It's not something I'm going to test you on, but if you're ever looking at a figure of DNA and you see a pentagon, that's pointing out the deoxyribose or ribose. Okay, so as you can see, we have a lone hydroxyl group there. If you see a lone hydroxyl group, that usually means it's the 3' end. So this is the 3' end right here. So knowing just that, we're able to fill out every other end of DNA here. So if that's the 3' end on that end, this phosphate group right here must be pointing out the 5' end. Since DNA runs anti-parallel, so it's parallel and it's flipped. So anti-parallel, if that's the 5' end on the right, this must be the 3' end on the other side. Knowing what end is which on the left-hand strand of DNA, we know the other end must be the 5'. So in this case, the arrow is indicating the 3' end of DNA. So really here, if you just look for the hydroxyl and phosphate groups, if it's a lone phosphate group, that means it's usually going to be the 5' end. If it's a lone hydroxyl group, that means the 3' end. So that's usually the easiest way to figure things out. But if you ever forget, remember that carbons are numbered clockwise from the nitrogenous base. And depending on which carbon is free, that determines what end of DNA you're looking at. So please be aware of that for exam questions. So here's a little bit of a critical thinking question. So here's a figure I actually took from Google, and I want you to tell me what is wrong with the directionality of the DNA strands below. So there's something very wrong here that you should be able to figure out using the content we've learned earlier in this lecture. So as you may have noticed, these strands aren't anti-parallel. So if you look here, we have a phosphate group and another phosphate group on the other hand. So here we have two phosphate groups that are free, one on the left-hand side, one on the right-hand side. So both of these strands are actually 5' on the same end. As you guys can see, on the other end of the strands, we have free hydroxyl groups or free 3' carbons. So both ends are actually 3'. So again, we don't have anti-parallel strands here. So this actually should be flipped. So the right-hand side should actually be flipped. So we should have 3' here and 5' here. So whoever made this figure made a huge mistake, and they accidentally made their DNA strands non-anti-parallel. So again, this is just to show that DNA strands are anti-parallel. So if one end is 5', the adjacent end must also be 3'. So this is just to show that DNA strands are anti-parallel. So if one end is 5', the adjacent strand must be 3'. So this is just to show that DNA strands are anti-parallel. So if one end is 5', the adjacent strand must be 3'. So this slide right here talks about DNA versus RNA, which is something we talked a little bit about earlier in this lecture. So DNA, as you probably know, stands for deoxyribonucleic acid. RNA, though, stands for ribonucleic acid. So really the difference between DNA and RNA is this deoxy prefix right here. Other than that, they're very similar. So as you can imagine, deoxy is relating to the fact that DNA actually has one less oxygen in it. So that's called deoxy. So oxygen, of course, is a key word here. So looking at this molecule we have on the left, this is RNA, or ribonucleic acid. So it has a hydroxyl group on its 2' carbon. So the hydroxyl group, just a reminder, is the OH there. Deoxyribonucleic acid, or DNA, on the right-hand side, has a hydrogen there. So on the 2' carbon, you have a hydrogen instead of a hydroxyl group, or OH. So that's the major difference between DNA and RNA. On the 2' of RNA, you have a hydroxyl group. On the 2' of DNA, you have just a lone hydrogen. So if you ever forget the difference between DNA and RNA, just remember what DNA and RNA stands for. So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. So that single OH group actually has a huge impact on the structure of both DNA and RNA. So that hydrogen that you find on DNA makes a double-stranded helix much more favorable. So as you can see, DNA has a double helix. So you have DNA on the left. The hydroxyl group on RNA, though, makes a single strand much more favorable. So as we're going to see in the next lecture, RNA actually likes being single-stranded. So instead of having a double helix, we just have a single strand of RNA forming a helix by itself. So here, just be aware that even though it's just a small change in the nucleotide, so a small change in the sugar, they're actually having a huge impact on the structure of these two molecules. So we went into a little bit more detail about the sugar that's found in the sugar phosphate backbone. Now we're going to talk about nitrogenous bases. So these link adjacent strands of DNA. So nitrogenous bases fall into two major categories. We have pyrimidines and purines. And you can tell the difference based on the structure of each nitrogenous base. So pyrimidines are thymine and cytosine. The shorthand way of writing this is T and C. The purines are adenine and guanine. If you look closely, the pyrimidines form a single circle like this, and the purines have two circles. So just be aware of that. Purines are two-circled and pyrimidines are one-circled. There are, of course, much more complex ways to identify the difference between the two, but since this is not a chemistry class, I won't go into more detail. So there's a little bit of a difference between RNA and DNA and the nitrogenous bases they use. So RNA uses uracil instead of thymine. So thymine is not found in RNA, instead we find uracil. So as we talked about when we looked at the structure of DNA, we have nitrogenous bases actually hydrogen bonding together. And this gets a special name, so we call this base pairing. So the bases or nitrogenous bases pair through hydrogen bonding. And specifically, the base pairing follows this scheme: A to T, so adenine to thymine, and G to C, guanine to cytosine. In the case of RNA, where we have no thymine, instead uracil, it's A to U. And again, this is all based on hydrogen bonding. So as you can see, we have hydrogen bonds, which are these dots here, linking together these nitrogenous bases. So it's very important that you know which bases pair to which. So again, T to A and C to G. And here's what this looks like in an actual strand of DNA. So you can see we have the sugar phosphate backbone again. We have our 5' end, which has our lone phosphate group, and we have our 3' end, which has our hydroxyl group. Since DNA strands are anti-parallel, we know since we have 3' here, this must be 5' on the other end. And if we go further than the same strand of DNA, we have our 3' end over there. So here again, you can see there are hydrogen bonding in between these nitrogenous bases. So that's actually what links together the different strands of DNA. So now we're going to start talking about DNA replication. So this is actually something we've already talked about, just not in very much detail. So if you remember back to mitosis and meiosis, in interphase, DNA is replicated. So we've already talked about DNA replication, but now we're going to go into the molecular details of that. And as a reminder, here we have the cell cycle. We have interphase in blue. So interphase actually takes up quite a bit of time in mitosis and meiosis. And hopefully, as you remember, we have S phase of the cell cycle, and that's when DNA is replicated. So that's actually when we're going to see the process we're going to be talking about in the rest of this lecture. So remember, during S phase of the cell cycle, we see replication of DNA and the process we're going to be talking about. So that's just to link together the various concepts we've been talking about in this class. So I'm going to start off by showing you guys a video. The reason why I chose this video is because it actually is very complex, but also simple enough that you can understand it without knowing much about DNA replication. So it's definitely a video I would highly recommend you come back to later in this lecture, or maybe a few days later. Come back and rewatch it and see if you can understand more of the video. So it goes into the appropriate amount of depth, but it's not too complex, so even a beginner can understand it. So I really like this video, and that's the reason why I'm going to be showing it to you.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides. Because the two complementary strands of the DNA molecule are oriented in opposite directions and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
So now that we watched the video, here we're going to go into the step-by-step process of DNA replication. So first, I'm going to introduce this term here, semi-conservative replication. And this essentially means that each strand of DNA becomes a template. So here we have our parental molecule of DNA. So the two blue strands, those become pulled apart in DNA replication, and we have two new strands. So two separate new strands synthesized off of those blue templates. So the parental strands serve as templates, so this is semi-conservative replication. And it's called semi-conservative because each new strand has one parental DNA and one new DNA that's synthesized off of that parental strand. So this is semi-conservative DNA replication. So now we're going to zoom way out and we're going to be looking at the whole entire process itself. So in a parental DNA molecule, so it's just the DNA floating around in a cell, origins of replication start forming. So these are bubbles that actually pull apart. So they're literally called bubbles. Start pulling apart, and we have the process of DNA replication start. So as you can see, we have one strand go this way, another strand go this way, this way, this way, this way, and this way. So really the point here is that the cell is actually trying to speed up the process of DNA replication. So by having many origins of replications or many bubbles where DNA replication is happening, we can actually speed up the process of DNA replication. So eventually, those bubbles will just become larger and larger. As you can see, they go left, right, and eventually those bubbles will actually meet and those new strands of DNA will actually fuse together. So just as we saw in the last slide where I talked about semi-conservative replication, now we have 2 new strands of DNA, each strand having 1 parental strand and 1 new strand. So that's a big overview of DNA replication. Now we're going to start zooming in and talking about the specific enzymes or molecules that catalyze the reactions of DNA replication. So probably the most important one is DNA polymerase. So this is actually the enzyme that synthesizes new strands. So the next 2 points I'm going to talk about are extremely important, so you have to make note of this. So DNA polymerase runs continuously in the 3 to 5' direction on the template. I'll say that one more time. DNA polymerase runs continuously in the 3 to 5' direction on the template. So nucleotides can only be added to the 3' end of DNA. And that's the reason why it runs continuously on the 3 to 5' strand. On the 5' to 3' strand, so the opposite strand of DNA, it runs discontinuously. And that is, it forms short, unlinked strands of DNA. So 3 to 5', it runs continuously, 5 to 3' discontinuously. And this has specific names. So for the continuous strand, we call that the leading strand. So as you can imagine, if it's running continuously, it's leading DNA replication, so it's happening faster and is leading. The discontinuous strand, though, we call the lagging strand. Again, discontinuous replication is happening slower, so it's lagging behind the leading strand. So you're absolutely going to find questions related to these ideas. So if you remember what strand is continuous and what strand is discontinuous, so 3' to 5' is continuous, 5' to 3' is discontinuous, then you'll know which is which. So please be aware of this. And so here's a nice little image showing what DNA polymerase does. So as you can see, we have the 3' of the old strand here. It's kind of hard to see since this actually has a double helix shape, but you have the 5' on the other end there. So as I just said, the leading strand is a 3' to 5' template strand. So here you can see DNA polymerase is doing a very nice job of synthesizing one continuous strand. On the other hand, though, on this 5', the 3' strand, we have discontinuous replication. So you can see DNA polymerase is coming along and is forming short fragments. So these fragments actually get a special name. So we call these Okazaki fragments. So this is actually named after the Japanese scientist who originally found these fragments. So again, on the 3' to 5' template strand, we have continuous replication. 5' to 3' template, we have discontinuous replication. So DNA polymerase can only form short strands called Okazaki fragments. Next up, we have the enzyme primase. So it's important to know that DNA polymerase can't just start by itself. So it can't just automatically sit down on a piece of DNA and start replicating that. So it needs a short sequence laid down beforehand. And that is done by this enzyme primase. So primase primes the process of DNA replication. And more specifically, it lays down a short RNA primer, or essentially a short piece of RNA, that allows DNA polymerase to start working. So again, DNA polymerase needs a short starter sequence, or primer, the technical term, to start the process of DNA replication. So that is done by the enzyme primase. So here's a nice little figure that shows that process. So here we have the 3' end here, and you can see that primase lays down a short primer there so the DNA polymerase can start working. It's also important to note the direction of this primer. So the primer itself was anti-parallel. So you can see that we have the 5' end here. On the other strand of DNA, we have the 3'. So the next step, as I just said, DNA polymerase comes along, you can have elongation of the strand. So it's important to note that for each of these Okazaki fragments that we talked about, each one of those Okazaki fragments needs its own primer. So each time DNA polymerase starts, it needs primase to lay down a primer. And as you can imagine, this is a very complex process that requires quite a bit of time comparatively to the leading strand. And that's one of the reasons why the lagging strand is much slower than the leading strand. Again, because you need primase to lay down primers for this process. It's also important to note that the leading strand itself also needs primers. So it's not just a lagging strand thing. The leading strand itself also needs primase to lay down a primer for DNA polymerase to start. So the next major enzyme is helicase. So DNA is double-stranded and usually tightly wound. So as you can imagine, it forms those helixes. Also, of course, hydrogen bonds are binding together these strands of DNA. So helicase actually unwinds or unzips the DNA strands. So it pulls apart those hydrogen bonds and allows for the DNA strands to separate. So it actually sits at the very base of the replication fork. So this whole entire thing right here is a replication fork. So it sits at the base and uncoils and separates the strands of DNA. There are also other parts of this figure that I like. So it talks a little bit about DNA polymerase. So you can see it's forming the leading strand. You can see there's also primase there, ready to prime any new synthesis that's going on. In the next slide, we're actually going to talk about ligase, which is the last enzyme in DNA replication that we're going to talk about. In this replication, remember we form short Okazaki fragments. So in between those Okazaki fragments, the phosphate sugar backbone is not actually covalently bonded yet. So DNA polymerase can't actually do that. It can't form the phosphate sugar backbone in between those Okazaki fragments. So really the point of DNA ligase is to create that sugar phosphate backbone. That's exactly what it does here. So you can see we have a hole right there. So the sugar phosphate backbone is all linked together. You can see ligase coming along and is bonding together those Okazaki fragments. So I should say that between the Okazaki fragments and the template strand, there is hydrogen bonds. So they're hydrogen bonding correctly, just the sugar phosphate backbone is not linked together. So ligase, again, is the major enzyme that's going to link together the sugar phosphate backbone. And so here's a nice figure from your book. As you can see, it's not quite as complex as what I've just talked about. So in regards to depth for the exam, I want you to be able to identify the enzymes I talked about in the last few slides. But other than that, this essentially shows exactly the same thing I talked about. So in the 3' to 5' template strand, this is our leading strand. So you can see DNA polymerase is synthesizing that in one complete chunk. What's not shown here is primase. So remember, DNA polymerase cannot just start by itself. It needs a short RNA primer laid down by primase to start DNA replication. So DNA polymerase is just going to keep moving along the strand and synthesizing one complete strand. On the other hand, though, on the 5' to 3' strand, we have discontinuous replication. So you can see we have short Okazaki fragments formed that are eventually going to need to be linked. Also importantly, these Okazaki fragments, just like the RNA primers that we talked about, are anti-parallel. So you can see the 3' end of that Okazaki fragment is matching up with the 5' end of that template strand. In the bottom of this figure, you can see DNA ligase is coming along. So it's actually ligating together those Okazaki fragments. So again, it's linking together the sugar phosphate backbone. These Okazaki fragments in the template DNA, there are hydrogen bonds. So the point of ligase is just to ligate together the sugar phosphate backbone. One thing we didn't talk about, though, is the overall direction of replication. So that always follows the leading strand. So if you know what strand is leading, that tells you the overall direction of DNA replication. And that's again because the leading strand is faster, so it's going to be opening up or moving DNA replication in that direction. So that's it for this lecture. So again, I highly recommend you go back to that video I showed you and review that. So it goes over all the enzymes we talked about and explains it in a nice little animation. So it's a video I highly recommend since it's very helpful for your studies.